Bismillahirrahmanirrahim. We are studying the central nervous system, and in that we are studying the reaction of oligodendrocytes to injury. Oligodendrocytes, the seen as the myelinators, load the clear space around their nuclei. Much in the same way, myelin is washed from shown glial cells as well. The reactions of oligodendrocytes to injury, there may be three oligodendrocytes nuclei in the photomicrograph that are shown at the right small dark nuclei. And the oligodendrocyte nuclei are seen in the white matter among myelinated axons. Some of the nuclei in this field are astrocytes. Now, pathological reactions of the oligodendrocytes to injury are: oligodendrocytes swell in response to almost any type of toxic or metabolic change. Diseases with the primary involvement of oligodendrocytes result in disorders of myelin with demyelination or abnormal myelin formation. The two major groups of diseases affecting oligodendrocytes and myelin are the leukodystrophies, include inherited disorders of myelin metabolism and acquired demyelinating diseases, for example, multiple sclerosis. Then reactions of the ependymal cells to injury. These are the ciliated columnar epithelial cells lining the ventricles, do not have specific patterns of reaction. When there is inflammation or maldilatation of the ventricular system, disruption of the ependymal lining is paired with proliferation of subependymal astrocytes to produce small irregularities on the ventricular surfaces. They are called the ependymal granulations. Then certain infectious agents, particularly cytomegalovirus, may produce extensive ependymal injury with viral inclusions in ependymal cells. This is the ependymal cells, look exactly like ciliated columnar cells and like the ventricular spaced and choroid. Then reactions of microglia to injury. Microglia are mesoderm-derived cells whose primary function is to serve as a fixed macrophage system in the CNS. Mind you that the macrophage system of the CNS, that is called microglia. They share many surface markers with peripheral monocytes or macrophages such as CR3 and CD68. They respond to injury by proliferating in the inobulated nuclei rod cells as in neurosyphilis and forming aggregates about small foci of tissue necrosis. They are called microglial nodules and congregating around cell bodies of dying neurons, that is called neurophagia. This is the cluster of microglia, the macrophages of the CNS. Now we come to the cerebral edema. Cerebral edema, the normal brain weight is about 1200 to 1300 grams. The weight of the brain is important because in edematous brain it will be heavier, and that you can be identified that the brain is edematous. The brain edema may be vasogenic due to disrupted blood-brain barrier or intravascular because of intercellular and cytotoxic or intracellular. Cerebral edema, or more precisely brain parenchymal edema, is of two principal types. Vasogenic is caused by blood-brain barrier disruption and increased vascular permeability, allowing fluid to shift from the intravascular compartment to the intercellular spaces of the brain. The paucity of lymphatics greatly impairs the absorption of excess extracellular fluid. Vasogenic edema may be either localized, for example, adjacent to inflammation or neoplasms, or generalized. Then cytotoxic edema is an increase in intracellular fluid secondary to neuronal, glial, or endothelial cell membrane injury, as might be encountered in someone with a generalized hypoxic or ischemic insult or with metabolic damage. Interstitial edema or hydrocephalic edema occurs especially around the lateral ventricles when an increase in intravascular pressure causes an abnormal flow of fluid from the intraventricular CSF across the ependymal lining to the periventricular white matter. In generalized edema, the gyri are flattened and the intervening sulci are narrow, and the ventricular cavities are compressed. As the brain expands, herniation may occur. In the photograph, you can see the flattened gyri often signify edematous brain, and there is sulci are compressed against the calvarium. The production of and absorption of CSF, the choroid plexus within the ventricular system produces cerebrospinal fluid, which normally circulates through the ventricular system and enters the cisterna magna at the base of the brain stem through the foramina of Lishka and Magendie. Subarachnoid CSF bathes the superior cerebral complexities and is absorbed by the arachnoid granulations. This is the photograph of the interventricular foramina in lateral ventricles and third ventricle and the cerebral aqueduct, and showing the circulation of the cerebrospinal fluid. Choroid plexus, site of protection of the cerebrospinal fluid. You see that the choroid plexus is papillary in configuration. Hydrocephalus refers to the accumulation of excessive CSF within the ventricular system. Most cases occur as a consequence of impaired flow and absorption of cerebrospinal fluid. Only rarely is overproduction the cause of hydrocephalus, for example, with tumors of the choroid plexus. Hydrocephalus may be communicating type, is more common, which frequently is due to the presence of blood products causing obstruction to flow of the cerebrospinal fluid CSF in the subarachnoid space and absorption of CSF through the arachnoid villi. Or it may be non-communicating type of hydrocephalus, often caused by blood clot obstruction of CSF flow at the interventricular foramen, third ventricle, cerebral aqueduct, or fourth ventricle. This is the hydrocephalus. You see the dilated lateral ventricle seen in coronal section through the mid-thalamus. The frontanella closure is the key factor whether hydrocephalus will result in any cranial enlargement. See the hydrocephalus in the child. This is a dilated lateral ventricles in the hydrocephalus. This will be the presentation of hydrocephalus on MRI. Then raised intracranial pressure and herniation. When the volume of the brain increases beyond the limit permitted by compression of veins and displacement of CSF, the pressure within the skull will increase. Because the cranial vault is divided by rigid dural folds, the falx and tentorium, localized expansion of the brain may cause it to be displaced in relation to these partitions. If the expansion is sufficiently severe, a herniation syndrome may occur. There may be subfalcine cingulate herniation occur when unilateral or asymmetric expansion of a cerebral hemisphere displaces the cingulate gyrus under the falx cerebri. This may lead to compression of branches of the anterior cerebral artery. Or there may be transtentorial or uncinate medial temporal herniation occurs when the medial aspect of the temporal lobe is compressed against the free margin of the tentorium. With increasing displacement of the temporal lobe, the third cranial nerve is compromised, resulting in pupillary dilatation and impairment of ocular involvements on the side of the lesion. Then transtentorial herniation, the posterior cerebral artery may also be compressed, resulting in ischemic injury to the territory supplied by that vessel, including the primary visual cortex. Also results in Kernohan's notch and secondary brainstem or dural hemorrhages. The tonsillar herniation, displacement of the cerebral tonsils through the foramen magnum. This pattern of herniation is life-threatening because it causes brainstem compression and compromises vital respiratory and cardiac centers in the medulla oblongata. This you can see all types of the herniations. This is the subfalcine herniation and then transtentorial herniation and tonsillar herniation of the cerebellum through the foramen magnum. Dural hemorrhage involving the brain stem at the junction of the pons and midbrain. Then we come to the CNS malformations of the malformation may be of the neural tube, forebrain, posterior fossa and syringomyelia. In the neural tube there may be anencephaly, encephalocele, spina bifida, forebrain, poliomicrogyria, holorodoncephaly, agenesis of corpus callosum, posterior fossa, intratentorial and Horner-Shari intertentorial herniation, Dandy-Walker's cerebellar cyst or syringomelia. Next we will continue that in the next lecture, inshallah. Thank you.